I'm Dr. Jim Zoval. You're about to watch one of my video lectures. If you like this video and would like to get videos for your entire General Organic and Biochemistry course, you can do so at www.zovallearning.com. I'll give you more information about that at the end of this video. We talked about the monomers, the nucleotides. We saw a few cases where they're relevant in biology, just as the nucleotide, like for ATP, ADP, or cyclic AMP. Now let's look at the polymers that you get when these nucleotide residues connect to one another in a chain. We get nucleic acids, specifically RNA and DNA, polynucleotides. A polynucleotide is a polymer made from nucleotide residues. These videos are made to be viewed while using fill-in lecture notes. The pause in the audio is to give students time to complete the lecture notes fill-in. The lecture notes can be downloaded, free of charge, at www.zovallearning.com. Nucleotides bond to each other in a linear sequence as shown in this view graph. This diagram only shows 4 nucleotide residues. However, polynucleotides generally contain between about 20 nucleotide residues, for example in the smallest RNA particles, to about 250 million nucleotide residues, for example in human chromosome number 1. A chain of covalently bonded nucleotides is referred to as a strand. I'll elaborate on the bonding pattern in polynucleotide strands by first describing a dinucleotide. A dinucleotide is formed from 2 nucleotide residues. Nucleotides are covalently bonded to each other by a phosphodiester bonding pattern between a 3' carbon on one monosaccharide and a 5' carbon on the other monosaccharide. The phosphodiester bonding pattern is highlighted in yellow. As you've seen in a previous chapter, a phosphodiester bonding pattern occurs when a phosphate group is bonded between 2 other groups of atoms. In the case of polynucleotides, we call this bonding pattern a 3' 5' phosphodiester. This is because the phosphate group is between the 3' and 5' carbons, as shown in this image. Remember, we saw a bond responsible for polysaccharides. That was the glycosidic bond. We saw a bond responsible for peptides from amino acids. That was the peptide bond. For nucleic acids, the bond that makes the polymer is the 3' 5' phosphodiester. Now you work on a problem. Starting with the dinucleotide shown below, draw a trinucleotide by adding another nucleotide to the 3' carbon of the bottom nucleotide residue. When you're done with that, highlight the phosphodiester bonding patterns. You can use a circle to represent the organic base structures as I have done in the dinucleotide. Click pause and work on the problem. Starting with the dinucleotide, we add a third nucleotide residue to the 3' carbon of the bottom nucleotide residue. This gives us the trinucleotide. The phosphodiester bonding patterns are highlighted with yellow. You're looking at a model of the structural formula of a small section of DNA. I want to use this model to point out some more details of the bonding patterns in polynucleotides. Can you tell me by looking at this how you know it's DNA and not RNA? If you figured out that it was DNA because there are deoxyribonucleotides, then you're correct. The nucleotides are connected to each other by covalent bonds between the monosaccharide residues and the phosphate residues only. For this reason, the alternating monosaccharide and phosphate residues in a polynucleotide strand are referred to as the sugar-phosphate backbone. The organic groups branch off the sugar-phosphate backbone, much like side chains branch off from a peptide chain. You learn that the ends of peptide chains are different from each other and are labeled as the N-terminus and C-terminus. A similar situation occurs at the ends of polynucleotide strands. One end of the strand is terminated with a phosphate group that's attached to a 5' carbon. We call this end of the strand the 5' terminus. The other end of the strand is terminated with a hydroxyl group that's attached to the 3' carbon. We call this end of the strand the 3' terminus. Let's continue to discuss the structure of DNA. DNA contains the information needed for life. This information enables cells to grow and divide. It is responsible for your physical characteristics such as your height, skin tone, and eye color. Human DNA contains about 3 million deoxyribonucleotide residues. Eukaryotic cells, which are cells having membrane-bound nuclei, have most of their DNA in the nucleus and small amounts of DNA in organelles such as chloroplast and mitochondria. The DNA of prokaryotic cells is contained in the cytoplasm. Genetic information, the information used to make the various proteins and thereby enabling life, is contained in the sequence of nucleotides in DNA. The sequence of nucleotides in DNA is referred to as DNA's primary structure. As an example, consider the primary structure of the small section of DNA that's shown in this view graph. As I mentioned earlier, DNA is composed of a combination of deoxyribonucleotides that contain either adenine, guanine, thymine, or cytosine organic bases. Nucleotides are given a one-letter abbreviation based on the first letter in the name of their organic bases, as shown in this image. In order to describe the primary structure of a DNA strand, it's customary to list the nucleotide's one-letter abbreviations in the order that the nucleotide residues appear in the strand. For example, the primary structure of the DNA strand shown in this diagram is written as 5' TGCA-3'. Another common method that's used to represent the primary structure of DNA and RNA is to draw the one-letter abbreviations of the organic bases branching from a line that represents the sugar phosphate backbone. I've drawn one of these representations for the DNA in the example here. In later sections of this chapter, you'll learn the details of how the sequence of nucleotides in a strand of DNA is used to direct the assembly of specific amino acid residues during the biosynthesis of proteins. DNA exists as a double helix, which is made from two polynucleotide strands. On the left of the screen, you're looking at a ribbon model of a DNA double helix structure. I'd like to draw your attention to a few important structural details that are seen in this model. Note that the sugar phosphate backbones for each of the two DNA strands are illustrated as the blue ribbons. The two DNA strands of a DNA double helix have their 5' terminuses and 3' terminuses with opposite orientations. Note that at the top of the double helix structure, the 5' terminus of one DNA strand is present, and the 3' terminus of the other DNA strand is present. The same is true at the bottom of the double helix. We call this opposing orientations of the DNA strands an anti-parallel arrangement. The two DNA strands are held together by especially strong hydrogen bonding between specific pairs of organic bases. Adenine bases hydrogen bond with thymine bases. Guanine bases hydrogen bond with cytosine bases. The reason for the strong hydrogen bonding between these organic base pairs is that they have complementary shapes. We say the adenine and thymine and the guanine cytosine shapes are complementary to each other because the positions of the atoms allow strong hydrogen bonds to form. The illustration on the left shows the hydrogen bonding between adenine and thymine and guanine and cytosine bases. The hydrogen bonding between these pairs of organic bases is referred to as base pairing. or as complementary base pairing. We say that the two polynucleotide strands in a double-stranded DNA particle are complementary. Let's work on a problem together. Let's find the complement to this short strand of DNA. The shorthand notation for the primary structure in this DNA strand is shown on the right. Its sequence is, in the order of the 5' direction to the 3' direction, TGCA. Because the two strands of DNA in a double helix are arranged in an anti-parallel orientation, we'll start with the 3' end of the complement on the left. The complementary strand is then built by using the correct base pairing. Where we see a T in one strand, we put an A in the complement. Where we see a G in one strand, we put a C in the complement. Where we see a C in one strand, we put a G in the complement. And where we see an A in one strand, we put a T in the complement. Now you try one. Fill in the missing one-letter abbreviations for the organic bases in the image below to show the correct complementary base pairing. Click pause and work on the problem. Check your answer. You can get your entire general, organic, and biochemistry course as a series of video lectures. The rental period is for 1 year, so you'll have these video lectures for the entire term of the course. You could watch the videos whenever and however many times you wish. I survey my students and they prefer these video lectures over the textbook by a ratio of about 10 to 1. Fill-in lecture notes and an electronic version of my textbook are available free of charge at the website. To get more information or to get the videos, free textbook, and free lecture notes, visit my website at www.zovallearning.com.